Yeah, so Music and Math is a group that came about talking with Catherine Lynn. We met her through Devon at an arm wrestling event. She got a PhD in mathematical theory of tuning musical instruments, which is super cool. Her partner works at a quantum computing startup, but mostly me and Catherine had this conversation. Her question was, is music hardwired into the universe? Like major and minor scales based on ratios, we perceive them with clearly different emotional tones. Are music and human emotion hardwired numerically into the universe? I suggested yes. This led to a follow-up question: what are the beautiful numbers in biology? Like in math, you have pi, e. In physics, you have fine structure constant, maybe cosmic microwave background radiation. Biology, we have fewer of these. So it just became a very open-ended, collaborative group. When we talk about beautiful numbers in biology, we often think of frequencies like brain waves. Michael Levin is causing a paradigm shift to look at bioelectricity. So we've been looking at that and looking at it from both, I would say, an evolutionary and mythological perspective. Little teaser trailer, basically serotonin and dopamine kind of feel like God and the devil, or female male in a way. There's something very deep there. Evolutionary mythological perspective, and then an actionable perspective, looking at gap junctions, looking at specific frequencies. Super fun, integrating work from a lot of different authors. So we're going to jump into that tonight.
Yeah, that sounds good. Onward. Sweet. Also, and this hasn't been included or anything, but as far as the fundamental numbers in biology, so the allometric scaling laws, I'd love to show you a little bit more about those.
Yeah.
They're basically all in ratios of 4 to something, 3 to 4, 3 to 4, 1 to 4.
I did look them up.
Sweet.
Yeah.
What did you think about those?
What I gathered was, what I gathered so far was it looked like they would be expected to be 2 to 3, but they go as high as 3 to 4, was what I gathered. Like 2 to 3 would be expected, but 3 to 4 is the upper end of the range.
One of the ones that stuck out to me when I was looking up at these allometric scaling laws are like basically plotting the logarithmic mass of a certain species versus some property like its heart rate. So the reason you do log scale is because you have organisms that are on the mass of grams all the way up to thousands of kilograms. So you're not going to plot that on a linear scale. You're going to have everything kind of bunched up together weird. So you plot it on a log scale, order 0, order 10, order 100, order 1,000. In any case, when you plot on these log mass versus the log heart rate, you get you get lines, which is surprising. Lines of like slope -3/4 or something.
Yeah.
And one of the ones that sticks out to me that I recall, if I'm recalling it correctly, is as the mass of the organism goes down, the heart rate goes up, right? So like an elephant has a slow heart rate, these whales have slow heart rates, but like a mouse has like really fast heart rate.
It's related to this. So across animal species, lifespan and body size correlate like very directly.
Right.
Bigger animals live within a species, bigger animals live less long. So like bigger dogs live less. The only exceptions to this are generally either flying animals or underground animals. So like bats, birds, and then like naked mole rats. Basically extreme animal, but the flight in particular I'm obsessed with because it's the mitochondrial energy generation that is or in evasion of predators, but also mitochondria. But
you also have like arterial diameter scales logarithmically with their mass. Yeah, like you look at their average diameter, it doesn't go up proportional. It goes up like to the power of either a fourth or a 3/4 or something. And the one thing that's amazing is when you look at these heart rate versus mass versus heart rate, right? But then you look at mass versus lifespan. Lifespan often goes up with mass. Right? Mice have like these short lives, and then when they have the same relationship, it's like an inverse relationship. So that if you multiply their heart rate by their lifespan, it actually comes out to be the same constant number. So each of these mammals gets the same number of heartbeats on average in its lifetime.
What's the number?
It's a number. I'm not sure what the number is, but you
We
just
get-
have to take it. Yeah.
We got to define the
It's
number.
like it's not like not a billion, but it's like
I
something.
think it's around order of magnitude a billion, I think.
Yeah, yeah.
I,
Every mammal gets a billion. Doesn't matter if it's a billion in 2 years or a billion
so
in
this
100
touches
years.
on a very deep question, and there's one paper in particular I want you to be aware of, but it goes back to the 1920s Raymond Pearl rate of living.
Okay.
This was touched on in the early 90s by like Van Flateren and maybe one other group. They used DAF2 mutant worms. It's the emergent property of vitality in living systems. To my gut, what you're saying is suggestive
Yeah.
of an emergent property of vitality. Couple things, but then mid 2000s Harvard Systems Biology lifespan machine, again, they got way bigger data than ever before using worms and like a 6 square foot scanner bed, just constantly measuring worm lifespan. And they said you can only contract or dilate time. Basically, vitality is an emergent property. They did say there were a couple genetic interventions that could change the shape of things. One thing to think about is life on Earth is all Model T Fords, okay? The same engine of iron sulfur that evolved on hydrothermal volcanic vents on the ocean floor 4 billion years ago is the same thing in our mitochondria now. We've never built a Tesla or even a Prius. Like, it's just the same. Iron is the thing that our engine's made of iron, and iron is and oxygen, which the engine runs on. Those are the things that slowly kill us. It is hard to change out the engine, but it's totally doable. This emergent property of vitality, I talk about, possibly wrongly, but cold fusion metabolism. You want low input, high yield, clean burning metabolism, because life is energy to overcome entropy. How do you get more life? You need more energy with fewer wasteful byproducts. What do you think about this? Anything in this cluster?
Yeah, I mean, like this touches a little bit on the laws of thermodynamics, where when you have energy that you're using to do useful work, you're going to have a byproduct of heat, which is not useful work.
Okay.
This is a necessity. If you're going to be inputting energy into a system to do some kind of ordered work, the necessary byproduct is an exhaust. It's an exhaust pipe.
Really?
That outputs heat. Yes, it's a law of thermodynamics.
I didn't know this.
Yeah, and you measure the heat output to the work done. I might have these numbers wrong, but it's essentially like the efficiency of the engine.
Well,
And a perfectly efficient engine, which is like a zero entropy, it doesn't exist, right?
But how close can we, what's the range of possibilities? How good are we at this? And why is it not applied to biological systems more?
It's a good question. That's a good question to look into, to
Okay.
think more about in the biological context.
Yeah, so one thing of interest, so mitochondria, the notorious fat burner DNP, it works by, the way metabolism works, you chop stuff up into vinegar, steal its electrons, give the electrons to NAD, NAD hands the electrons off to an iron highway, and that creates a proton gradient. The protons flow up through a spinning motor that makes ATP. DNP steals the protons, and you churn through calories and you make heat instead of ATP. There's a genetically encoded protein, uncoupling protein, that can also do this. So it is literally heat versus energy. In biology, a lot of the time, there's like a heartbeat of stress in cells that a little bit of oxidative stress, a little bit of uncoupling leads to more efficiency long term. This is deeply true, but I also think it's something that can be overcome eventually, just like a perfect system, but food for thought. Yeah.
Yeah, I think that 1 billion heartbeat for all these organisms is a very there's a deep explanation behind that with this counting. It's like an integer count. Like you're counting, and there's some sort of that's always struck me as a pretty deep insight into fundamental mechanism.
Very.
And then there is this heat exhaustion, entropy increasing nature of simply doing mechanical work. That's it in every single machine. You just work out, you get hot. That's your cells experiencing this exhaust.
I want to think about this. Okay, so because you have the physics perspective on it. Okay, so the limits of human metabolism are really well defined. Okay, exercise cyclists in particular is highly quantifiable. like wattage output sustained over the course of an hour,
Yeah.
VO2 max, even not adjusted for body weight.
Yeah.
There's really, really hard limits. So like 7.7, whatever it is, like liters per minute
Watts per
of
kilogram or
oxygen.
something? Watts per kilogram?
Not normalized to body weight, because we want to just look at like overall bioenergy, I think. But the same guy holds both the non-adjusted for body weight and the adjusted for body weight records. The Norwegian Christian Blumenfeld just broke 100 adjusted for body weight, 7.7 not adjusted, but you can hold like 450 watts for an hour on a bike. They're super well defined.
Yeah.
And I think if we can like, there's a number of crazy things. So it starts with the gas. We use a quarter of the oxygen we breathe, which is nuts. Like how you can get away with that level of inefficiency to start with. But then you get to the mitochondria. I mean, you could make more mitochondria. You could mop up dangerous waste more. I think oxygen is typically thought of as the biggest limiter. I don't know.
Is it a wild tangent, but have you ever looked into the bowhead whale genetics? Because aren't they an outlier? They live like 200 years.
Well,
and
first of all, they're big.
Yeah. But they
you know-
essentially don't get cancer after 200 years?
Yeah, I mean, I don't know their genetics well.
Okay.
So there's a few things, Greenland shark. I think there's a few species. Rockfish also. There's a few species that live to 200 to maybe
Yeah.
200 to 400. Most of them live in very cold water, which is kind of a cheat code.
It's almost like an entropy reduction.
I think
thing.
there's one
Yeah.
rockfish that doesn't live in very cold water, and there's a dude from Steve Alsted's lab who was doing their genetics. So elephants I think have extra copies of TP53 tumor suppressor.
Okay.
I'm gonna butcher his name, but like Yao Pedro de Magales is one of the best comparative biology guys for this type of stuff. Short answer is no. I haven't looked at comparative genetics a ton.
I'd love to get your insight if you ever have the time, like if you ever have a tangent on the billhead.
I got a couple tangents real quick at
Yeah.
least. So for DARPA biostasis, we're looking at hibernation is metabolic depression. Humans go down to 85% during sleep. In the animal kingdom, or in other species, you get down to 50%, sometimes 0 to 10%. Intrinsically disordered proteins, I think in plants mostly, are one of the big chaperones that can protect against maybe freezing.
IDPs, is essentially what these regions that were my thesis, right? They had these IDPs in them. So I don't know if you call proteins with disordered regions in them IDPs, or if the entire protein itself has to be intrinsically disordered.
There were IDP proteins.
Okay.
We have chaperone proteins, and there are chemical chaperones like trihalose is the big one. But this is like this is a lot of this is like metabolic. You can get more energy, you can get cleaner energy, you can live in stasis. There's the genome, which is really one of the easiest things to correct. Like it's just a faulty code. There's so much low-hanging fruit there.
Okay.
What we're reaching out with Levin is like more spiritual. Like it touches on like chakras and like halos and
It's
So
more
what-
systemic, I would say. It feels like
Systemic-
tissue level.
just the overlay of electromagnetism on biology and light is like really underdeveloped. Like it's just not, even with smell, like there's this lock and key mechanism, and then there's this one group that was like, no, it's molecular vibration. Like, because you can have like two identical molecules, but one of them has deuterium, and they'll smell different. So it's the same
It's
lock.
like a resonant frequency
Exactly.
that
Yeah.
resonates differently. Okay.
Yeah, and this is beyond my.
That's interesting.
The first time I got the EM spectrum explained to me was my advanced chemistry friend who does like quantum dots or whatever this stuff is.
Yeah.
And he was like, walk me through it. But I don't like that type of vibrate. First of all, there's so many molecules. They all have vibrate, like
Yep.
so many numbers.
Of course. Yeah.
And then there's the electromagnetic spectrum.
My initial idea to look into some of these molecular frequencies that may show up, or even like the resonant frequencies at which different molecules can absorb light. My impulse is that the frequency itself may or may not be of interest, but the ratio of that frequency to some other fundamental frequency in biology so that we get a single number as opposed to something with units, right?
Let's talk about this for a second.
Yeah.
So, resonance is a word I highlighted this morning in my notes before we talked. I think I got it from I highlighted it twice. Maybe it's from talking to you. No, it's from an LLM actually. So basically, I don't know what this means. Faraday's law of induction.
Yeah.
Basically, with these voltage-gated channels, like if there's environmental light or EM or radio, because radio is just light, the non-visible light, right? If it matches the frequency of the channel or something in a resonant way, it can affect it.
I'll have to, yeah. I mean, well, first up, Faraday's law just tells you that if you change, so electric and magnetic fields are basically two faces of the same thing. The same fundamental electromagnetic sort of entity that exists, or sort of field. Faraday's law essentially just tells you if I change the magnetic field here, like if I have a magnet right here and I was like kind of move it around a bit, where that magnetic field's changing, it's generating an electric field around that change. So if you change an electric field arrow, or a magnetic field arrow, you shrink it, an electric field circulates around that changing. Now, as that electric field changes, magnetic field circulates around that change, and as that changes, it's a self-perpetuating process, and that's light, by the way. It's the self-perpetuation of you change B, and therefore E changes around that, but that changing E has a changing B around it, but that changing B has a changing E around it, and that propagates through space, and that's light. That's light propagation. It's a perpetual reaction to fields changing with each other, and it's like weird dance that goes on at the speed of light.
That was way over my head. I got lost on that one.
It's all good. But Faraday's law of induction is that you change a B field, which is a magnetic field, you generate an E field. So one thing that is of interest, possibly, B fields are very weak, by the way. B fields are weaker than E fields by a factor of the speed of light, which is a large number. So, but one idea you could have is that if you have a membrane potential and you suddenly introduce an electric field, around that electric field has to be a magnetic field that is going to be basically coming to existence by Faraday's law. But now whether that magnetic field can be biologically relevant is a whole other question. But we can't really rule out you can't immediately rule out the importance of the electric field. Actually, if I'm not mistaken, it's pretty damn big, because it's like a 0.7 millivolt voltage, but the distance between that is like something on the order of nanometers. And if you want to get a rough estimation of the electric field strength, you take the voltage divided by the distance. It gives you a rough estimate. It's a pretty big number. So it could generate a pretty significant magnetic field, which may act on the spin angular momentums of nearby particles. Who knows? Nearby molecules. I don't know. But there is, of course, precedence for magnetic importance of magnetic force in biological systems. Namely, I mean, magnetoreception is a real thing. Birds navigate. Turtles navigate.
Is it?
Yeah, because they can chop out parts of.
Exogenous, right?
They can remove parts of their parts of their beak, I believe, and they'll no longer be able to navigate in the magnetic field of Earth. They can change something.
Really curious about that.
Yeah, my last one.
They can, yeah.
My last impression of that. So there was a guy in our lab in Seattle who was from here in Austin. There was an Austin lab that I think established the existence of magnetoreceptors first in worms.
Okay.
And he was basically like, yeah, their research is going to be hard to replicate or something like that. I don't know.
Okay.
And then I've heard about what you're referring to, but I don't know the literature well at all.
I'm aware that they took, if I'm remembering it correctly, they took a part of a beak, the front part of a beak out of some sort of type of bird, and it could no longer navigate its way in back to its, it was like it could no longer navigate its way along the Earth's magnetic field in the way that it did before.
That's compass, though. That's not magnetic field generation.
Okay.
That's not magnetic field generation.
Not generation, but it's responding to the strength of the Earth's
look,
field.
There's a paper. So we recently had the opportunity to co-publish with ions on a mechanism of telepathy. One of the papers we cited is this paper by this Iranian guy, kind of out of left field, all right?
Okay.
And he reviews potential mechanisms for brain-to-brain communication. I think he went
Okay,
into
that's
some of
fine.
this stuff because there's like radical pairs and like magnetite.
Okay.
I'm gonna find his paper real quick because it was really. I feel like it was really good and it was really unique, and I feel like it's related to this. But it's I don't think it's the
sounds
original
fun.
research like on the bird beak stuff.
Yeah, but it was just this idea of like brain to brain communication.
I think it was just a good paper reviewing potential mechanisms of such.
Yeah, I think also one thing that I don't know if you agree with this at all either, but using telepathy is it's a cool word. It's like a fun word, but maybe I don't know how you define it, but I think whatever some people legitimately think of as telepathy it's going to end up being some kind of like folk word for like a real physical thing that people are doing, right? But it's just a folk word.
I don't think of it as a folk word. It is a real physical thing of brains just communicating with each other without. Yeah, that's what I mean by it. Like just like brain-to-brain communication at a distance.
I've just heard it used in so many different contexts.
Yeah, it's like
telepathic.
saying. I mean, we could say NHI instead of aliens, but like.
Yeah, we say
mean,
UAPs now.
Brain-to-brain
instead of, like,
communication is, to me, I'm a informal guy. Like telepathy.
Yeah.
Is what I say, and it's fun. But yeah, I mean,
communication in the absence of like obvious physical senses and being involved, right? Like hearing, sight, smell.
Like cold reading is real too, and that co-factors into telepathy in my personal experience.
Yeah.
So it's always important.
There's like weird people get gut feelings about like something bad's going on, but this could all be a sensory. Like you could be, who knows if people give off molecules that are so small when you smell them.
Skin.
I think the microbiome might mediate gut feelings and like certain types of psychic stuff because you're covered in like a gazillion non-human organisms that are.
Yeah.
And they're like, God knows what they can sense and transmit and stuff. We just act like our genome is the only one that they all have their own genome.
I don't know if this is a fact that's like commonly shared, but like, or commonly like dwelled on maybe, but like you said, we have a skin microbiome, we have a gut microbiome.
Tube and a tube, outer tube
Yeah.
and inner tube.
If you count cell for cell, how much prokaryotic cells are in us versus how many eukaryotic,
way
there's
more.
way more prokaryotic.
Yeah, way
so
more-
we're
way more
actually just like vessels for prokaryotic single cell.
I mean, by cell count, yes. By mass, we're squarely in the lead.
Yeah, just it's interesting by cell for cell. There's
Yeah.
more prokaryotic
Way more of them than
material
there are of us on us.
in us and on us.
And they don't obey any rules. Like when they meet each other and shake hands, they exchange genetic material. Like there's no defining them.
Okay.
They evolve. This is what people say. They're like, oh, because the human genome does not evolve that rapidly, but your microbiome genome evolves super rapidly.
Oh yeah, dude. I mean, people get like they change an entire like a dietary system
Yeah.
plan, right? They go keto, they go carnivore,
Yeah.
they go carb,
The
sugar.
100 trillion
Yeah, your gut
microbiome
microbiome
cells
goes
are definitely
insane.
evolving in
Yeah,
real
yeah.
time to that. But important distinction.
Yeah, people on high sugar diets, their gut microbiome is like this different composition, right?
You know.
So this has been gold, really. There's been like 5 or 6 really great
Awesome.
clips. Did you think about starting a new video? Did you want to get focused on music and math? That's where
Oh, shit.
it started.
Oh my God, we didn't.
I feel you lost the plot.
Yeah, I think you're right.
So
I shouldn't be too much involved in it, but I mean, I can talk about it.
You gave an intro to music and math that I can still use, but you said there's a big breakthrough. I didn't hear anything.
Yeah, we gotta create it.
You said there's something to report. So
If
No,
I'm not-
I
if
mean,
I'm- yeah.
no. Like I feel like we're close.
for what?
A breakthrough.
How in what? What's the question that you're asking that's being put?
I just know it when I like.
Alright, you said you had something specific.
I just have that good lucky feeling. Michael Levin is just white hot right now in biology. Like easy to make the argument that he's the smartest biologist alive. But the question is, what is he saying? I would say, simple way to put it, he's creating a paradigm shift where we're looking at bioelectricity as something that can be tinkered with in a therapeutic way. So, okay, what does that mean? Does it mean you just hook yourself up to electrodes? What it seems to mean is he's manipulating the expression of gap junctions. So neurons have ion channels that let electrolytes in to fire, and they have ion channels at synapses. Non-neuronal cells have gap junctions, much fewer gap junction genes, but gap junctions connect non-neuronal cells and allow them to exchange basically soup contents like electrolytes, other things, to all be on the same team. Like all your liver cells kind of need to share a little bit of their fluid to like be aligned. So he's manipulating, and these gap junctions create electrical potentials. I think I'm going to botch this language, but similarly, he's saying non-neuronal cells are a lot more like neurons. Like every cell's more like a brain than we realize. But so if we're looking at things to, the other thing he's had success with is serotonergic drugs. So, okay, gap junctions, I have a list of them. Like they're easy to manipulate, cool. But what about this serotonin business? What about other, when we think about electricity and serotonin comes from tryptophan. Melatonin is also in this family, so is DMT. They're all just like siblings. Very ancient. Dopamine is also in this pathway. I'm just becoming fixated on this question of what is the meaning here? Like, why did serotonin and dopamine evolve? And basically, dopamine, tyrosine are, melanin is a chain of tyrosines. Redheads make defective melanin by inappropriately inserting a cysteine every so often. Life started on the ocean floor probably with the iron. Iron was the first electronic highway. Then you kind of float up to the surface, start doing photosynthesis. I think this is when you get melatonin at night to repair. Melatonin is a mitochondrial, it's like the ultimate antioxidant, and at night it turns on, it goes in and cleans up the mitochondrial damage that occurred due to daily metabolism. So it's probably an antioxidant response, and then tyrosine and dopamine are probably like daytime UV protective response. There's some, I mean, one is rest and digest, one is fight or flight. There's something about them very deep, and they're both these aromatic rings, like, again, lining up. Tryptophans is also maybe part, tryptophans may be the seat of consciousness on microtubules. They might also be create quantum tunnels in complex 1 mitochondria. Like there's something about tryptophan, like adenosine, that is, and again, adenosine and tryptophan have the same shape. And then dopamine and the other DNA bases also have the same shape. There's something here, I can't see this pattern and not think there's something there. But right now, all it is a pattern. I want to define it in terms of its story and like say something new about it. One way we can get at it is what are the numbers, like what wavelengths of light do tryptophan, serotonin, melatonin, DMT, dopamine, tyrosine, phenylalanine, adrenaline interact with?
And then maybe this gets to the music and
what
math.
are the resonant frequencies of tryptophan, tyrosine, serotonin, dopamine? What are their evolutionary story arcs? So what mathematical and mythological meaning can we assign to these two players, and then also to similarly major players like adenosine, phosphate, iron sulfur clusters? It's a place to start. That and building out what the different gap junctions do and how they're harnessable. This will give us a much more actionable understanding of Michael Evans' work.
Cool. I mean, I started in particle theory, and then when I got to my program, the projects available in particle theory were actually pretty, unfortunately, they weren't really exciting. It was like you would put something into a machine and crank the machine and then get your PhD would come out of it by exploring these little things, these little properties of hypothetical particles in a parameter space, and that just wasn't really that exciting. So then I started looking around other departments, and that's how I found biophysics. And so you're asking like, what's my experience in biology? Dude, before I started like working with this guy, I really didn't have a good definition of what a cell was, right? And so, but I was like, he's gonna be asking me to be doing research on biomolecules. The question we wanted to address, the overall question was, why does nature encode proteins, like some 30-something% of our genome codes for proteins that have disordered regions in them, which means inherent lack of structure. They don't take on a three-dimensional configuration that's fixed. So in other words, what they're kind of doing in the cell is they're like jostling around like a dumbbell or like a tether, like a rope, like a flexible tether. And cells are immersed in fluid, and those fluid are undergoing fluid dynamics, which are dependent on temperatures. And think about if you have like what's the most likely, you have like a long cable in your house or like a long string in your house, the most likely configuration after a bunch of getting knocked around a bit is it jumbles up into a knot, right? So polymers tend to clump into these knots when they're allowed to explore their configuration space, and that's what temperature allows you to do in the thermodynamics. It allows you to explore the configuration space of that flexible polymer, so that's no fixed structure. And our fundamental question we want to look at was, why is nature evolving these flexible regions? What function are they playing that stiff proteins with fixed structures couldn't do? And we were reading a paper one day where we were looking at these things called foramins, which are a lot like microtubules, and that they grow. The foramin proteins will grow certain types of actin monomers, right?
F-O-R-M-I-N?
Yeah. So there's actin, right? Actin polymers, and you grow actin by adding on a monomer and adding on a monomer. And the question is nature sometimes wants to tune this up. The cell wants to tune this up, tune this down, slow it up, slow it down. And one thing that's helping these actin monomers grow is this foramin ring that sort of sits at the growing end, and it has these tether-like hands, and they kind of grab like these monomers. They kind of like grab and they kind of build the protein up, and the protein kind of like shuffles at the end, right? Yeah. And there was an interesting experiment done like some years ago where they actually they looked at the polymerization rate of an actin polymer as a function of force pulling on the string. So to say that again, they pull on that little arm, that little flexible arm that's supposed to be grabbing the monomers, and you pull on it, and you're trying to see how fast the actin grows. Okay, intuitively you would think, okay, the thing grabbing the monomers it has to deliver them back. If you pull on it and you stretch it out, it should just be an obvious curve. It goes straight down. Well, they saw something weird. They saw a temporary increase in the speed of growth, and then a decrease. So that's very strange. That's temporary speed up is very weird. Like, why, if these flexible arms are used for sort of capturing the monomers, how is it possible that you're speeding up the growth by stretching them out? And my answer, which was my thesis, was that maybe because the configuration of a polymer is generally knotted up, that's its natural configuration under no force. And when you start to apply force to a polymer, what you actually do is you stretch it out into a less likely configuration, but you're applying force to it, and what you're doing is you're opening up binding spots along the polymer, and that's what's happening. So my guess was that if you pull on this little arm, you're actually letting it catch more monomers, but you haven't pulled on it hard enough that it can't deliver them back to their growing end. So you increase the probability of capture by pulling on the protein, and you also don't inhibit the delivery too badly, and so you're in the perfect regime to accelerate growth. But then once you pull on it enough, you're just never going to deliver the monomer back, right? If it's just stretched out that much. So the intuition is correct on the far end of the force, but on that little small intermediate region, there's a trade-off. And the point is that nature's probably using forces and mechanical tensions to control and modulate its signaling, knowing that it's going to get a speed up or a slow down based on the architecture of the protein binding sites, and knowing that you're going to get occlusion in a thermodynamic environment. So there's an example where just the pure molecular mechanics are being overridden by essentially just the pure thermodynamic mechanical considerations. One really satisfying thing about this model, by the way, was that there are different classes of foramin proteins that have their binding sites at different locations and different lengths apart. And so with the model, we were actually able to do something which is rare in biology, is have a model that explained the experiment before us, explained it like perfect, it was like amazing, and then have a model that predicted as yet undone experiments on other types of foramin proteins. And as far as I'm aware, the latest result was that they pulled on a certain type of protein whose binding sites were arranged differently, and they saw the same prediction as ours, which is no speed up, only slow down. So certain foramin proteins are used for certain things, and that's the hypothesis, still working hypothesis.
Cool.
So